So hello everyone. Thanks Lauren. Thanks Ed for the presentation. I hope you can hear me. I had a problem with my voice yesterday. Sound a bit better today, but if you notice that it's too quiet or not clear enough, please let me know. I'll do my best. So hello everyone. My name's Serge Caston, and I came here from Abitep. It's a company that belongs to a bigger group. The group is called Andermatt. They're from Switzerland, and they specialize on biological solutions. And I was asked to prepare two presentations, and the first one we thought makes more sense to start with, the general biology, to give you, let's say, a broader understanding of the products we're going to talk about at the later stage. And I wanted to also give you a small insight into fermentation, because that's really what Abitep does. And also, of course, the mode of action of microorganisms in general. At Abitep, we're specialized in bacteria. So we are specialists of liquid fermentation. And I hope after this talk you'll understand more what that means. So let's dig into it. So first, I wanted to introduce myself a bit because I think most of you don't know me. I think most of you knew some of the other presenters because they probably come here annually. I think I really love the excitement and the interest about all of these topics here. I already learned a lot, and hopefully I can come here again next year as well. I love the presentation by Glenn as well, fantastic. So first, a little bit about me. So I'm a biologist. I have two big passions in biology, mostly. If I would have to choose, it would be marine biology and plant science. I love both. So I was really lucky at the university that I studied, that in my bachelor's I was able to do marine biology, and then in my master's I was able to concentrate on plant science. And at that time I was really into how can you grow plants to the maximum potential. And I was trying to find out how it can be done. And I was also looking into how, for example, the people that go to these competitions that grow, these huge pumpkins, huge cucumbers, any type of vegetable, there are competitions for that. And these guys must know, right? They grow these massive yields, or they produce these massive yields with these vegetables. And I wanted to know, okay, how do they do that? And I found out that it's not that they use a ton load of chemicals. They actually, if you investigate, they work a lot with nature together. And they use things, for example, I think the device over there is a compost tea brewer. So they use things like that. They use compost teas. They use lots of amendments, try to get like a healthy living soil. And that's how they produce these impressive yields, and break records year after year. So I thought that was super interesting. And at that university, we had a lab that was focusing on pathogens, or let's say plant microbe interactions, but mostly on pathogens. And I asked them back then if they're also working on beneficial, microorganisms, because it became, more and more of a hot topic. So, and they said, yes, we were just starting a new project and we're looking for someone who wants to do his thesis on that. And I said, yeah, great, here I am, let's go for it. And I was quite lucky to do that. And we looked at, different kinds of rhizobacteria, and from different groups. And we found out that most of them promoted the growth. And I mean, we selected them based on, knowledge that they were actually in the pool of the rhizosphere, so they were known to be associated with the type of plant we were investigating. We were looking at Arabidopsis plants. That's like a model organism in plant science. Yeah, and that was really, eye-opening for me, and it started to make me really believe into the power of microbes and what kind of effects they have with plants. And not only did I see, the growth promotion, but I also saw that they can increase the resilience of the plant against pathogens, which I think is also a very important aspect. And if you think about all of these things, then you can understand that, that there can be ways how to farm, or perform agriculture in a different way, working more with nature than against it. And I think in the last, couple, probably in the last century or so, we moved into a way where it wasn't really with nature, but more against it. So, and I think we're now understanding this more and more. I think the consciousness is going into the right direction, and we're understanding that there are other ways to do it a bit better. Yeah, so there's a picture of me inoculating plants and doing that at the university. And then while I was at the university, I worked at an innovation lab where I got into indoor farming. That set me off a trail where I worked at a company which was doing indoor farming. They were actually also moving out here to Canada. They had plants in Calgary and in Hamilton near Toronto, but also other plants worldwide. In the picture below, you see one of the bigger, large-scale vertical farming units. And that was really exciting. I worked there for 4 years in different kinds of roles. And then that company struggled financially at some point, and then I moved to Abitep, where I'm now since August 2023, so 1.5 years roughly. And I lead the product management team there, and since October I also lead the product development teams. So I have a lot of stuff to do at the moment. I'm spread quite thin, you could say. But it's really exciting, and I really feel that now, for me, really it has sort of closed a circle again, because now I'm back to where I came from when I got off the university, or the topics I worked at with university. Let me go into, let's say, the general biology for a bit, and I wanted to keep this, very simple. I think all of you have, let's say, a broad understanding of what's happening in terms of biology and, the overall chemistry and the physics and everything. But let me just go through it. So the primary source of energy is, of course, the sun, right? So the sun projects energy towards Earth in sun rays and in light energy. And those photons are picked up by plants, right? They convert that light energy into carbohydrates, into sugars. And you have this flow of energy in one direction. And so the sun rays hit the plant, and the plant converts all that energy, and heat is radiated. So you have one flow of direction of energy. But there is also a cyclic process happening, and that's the chemical cycle. Because after the sun's energy is harvested from the sun by the plants, they actually fix this energy, right? So they're actually laying the basis for all other life forms on Earth. So you have this chemical cycle where when the plant decomposes, and also the plant, of course, you heard it before by Glenn as well, the plant releases lots of exudates into the soil to feed the microbes. That sort of kicks off the first step of this chemical cycle. So the microbes then decompose the plant debris or other organic matter, of course, dead animals, everything that accumulates, and they make those nutrients available for the next generation of plants. So there you have, let's say, a chemical cycle and a balance of the ecosystem. What, of course, can happen is if you have a problem in this balance, or if the soil health is disturbed, right, then you don't have this chemical cycle happening correctly. You'll have maybe not the, you maybe have soil compaction, you won't have oxygen in the soil, you maybe have then therefore no life in the soil or not enough, so they can't really complete this cycle anymore, and the plants will really be affected by that. So I try to humoristically add a sort of a cheeky product placement here, but the chemical cycle can be reestablished if we do the right things, right? Probably you don't know this yet, but I'll explain it also more in the second part of my talk, which will be after lunch, where I'll tell you about the products we have. So this is just about how you can also get that balance right again. And I think you will hear a lot from also the other speakers on how you can do it. But yeah, there's a way to do it. And what I'm going to talk to you today about as well is how these processes can happen. So one of the things how bacteria, for example, or other microorganisms as well, work is that they mobilize nutrients for the plants, right? A lot of the nutrients in the soil are not really mobile, right? They're a bit locked up, and the plant needs the microorganisms to make these nutrients available. So one of the examples is iron, for example. Iron is a very, you have it a lot in the soil and in nature, but it's not in the right form that the plant can take it up. So the plant needs these microbes to produce what is so-called siderophore. And these siderophores, they make it possible that the iron is then reduced, and then you have a iron form, it's Fe2+, that can then be taken up by the plant roots easily. And that's a similar case with lots of the other nutrients. We heard before what the very important nutrients, calcium, phosphorus. So all of these things, nitrogen is also very important, as you know. And so how do the microbes actually do that? And there are three main ways how the bacteria, for example, can do this. So one of them is phosphatases and phytases. So these are enzymes that can hydrolyze organic phosphate, and the phytases degrade phytic acid. So those are the first main ways how the microorganisms can do that and increase the available phosphorus for the plant. Then they also release organic acids. So there's a variety of organic acids the microbes release, and they can really dissolve the mineral-bound phosphorus. Then what happens as well, that these acids, they can chelate the metal ions, and then they free up phosphates for the plant uptake. So they increase the available phosphate in the soil by just chelating these metal ions, because before they were bound up with phosphate, and now they're freed up so that the free phosphate is then available. Very similar, the siderophores I mentioned before that help also with the iron uptake. They also make sure that the phosphate pool is increased and you have more phosphate available. So those are the three main ways how bacillus species can increase the phosphate and make it more mobile for the plants. So now I want to show you how the bacteria colonize the root. And you probably heard about the rhizosphere before. It's just like the space of the zone around the roots. And I've gotten this question in the last 2 days as well, like, when is the best time to treat the plant, right? And I always say the earlier the better, but of course you have to have, let's say, also roots or a plant that supports the establishment of the microbes. So what is a really good treatment is, of course, a seed treatment. With the seed treatment, is very targeted. You just need a very little amount of product that goes on the seed, and it's very directed, right? It's right where you want it to be. And the other question I get is, why is this little amount sufficient to be able to be active in the plant? Like, how can that little amount we actually apply, per plant or per surface area, because it's very little if you think about it, how can that work? And the reason is that you only need a certain amount of spores at the seed, because the plant will help the bacteria proliferate and will feed it plant exudates, and the microbes will continue to grow. So you will find more microbes at a later growth stage than at the beginning. So that's why you only need a little amount at the beginning. So now I want to come to another big topic, which is more in terms of plant defense. And you probably know that there's an ongoing evolutionary arms race between pathogens and plants. And I just wanted to show you a few examples. So this poor tomato plant is affected by, or can be affected by, many different kinds of pathogens, right? You have fungal pathogens, you have fungal pathogens that go on the leaf, on the foliar tissue only. You have some that are attacking the roots. You have bacteria that can attack the whole plant. You have viruses, you have insects, all of this stuff, and the plant needs to be able to defend itself. And there's a constant evolutionary war fighting between these organisms, which I find fascinating. Some examples up here is just like, the plants have, of course, the first layer of defense. If you think about the foliar tissue, is the cuticula, right? The outside wax layer. It could make it thicker, or place certain metabolites inside this layer that maybe be antifungal or antimicrobial or things like that. They can add, extra layers beneath the cuticula to make it more difficult for pathogens to attack the plant. Thorns, for example, is also just a means of a plant to defend itself against predators. And I think this is quite ingenious, that some plants, by, of course, random mutation, have developed what you see here is an imitation of butterfly eggs on the underside of the leaf. So the butterfly thinks, okay, damn, this leaf has already been used by a different buddy of mine, so I can't. It's already taken, so I can't lay my eggs there. But it's actually fake eggs produced by a plant. This is just a picture of different kinds of pests attacking the leaf, because you can have bacteria, funguses like powdery mildew, botrytis, and so on. That's just another thing I wanted to show. So again, about the plant immune system, there are sort of three main ways, signal pathways, how the plant immune system works. And this is lots of knowledge from this came actually from my lab in Germany. And that's why I'm also showing this. So you could have, if we think about the systemic acquired resistance, so through different modes of attack, it could be through nematodes or through viruses, fungi, or bacteria. It's always a certain kind of pathogen that trigger this immune response pathway. Salicylic acid is increased. That's the first, plant defense metabolite that is increased in the plant, and you can measure that. And that sets off sort of a signal cascade. Of course, it starts with elicitors that come from the pathogens. It could be the fungus producing a certain chitin. Chitin can be an elicitor because it's typical for fungi. It can be, some bacterial compound that the plant actually notices at its receptors, and that sends off a signal cascade, and then salicylic acid is then increased, and then plant defense compounds are then also increased. So it could be that those plant defense compounds could be specific to that type of pathogen. So maybe the plant knows, okay, I'm being attacked by a mushroom or by a fungus. I need to boost up antifungal compounds. And I think that's also pretty interesting. Another thing that's pretty interesting is that salicylic acid can be methylated, and then what you get is a methylated salicylic acid, which can then move through the air because it can pass through the air, and that means the plant that is neighboring the plant that is being attacked notices, okay, my body here is being attacked by a fungus, so I have to start maybe boosting up my plant defense metabolites to be prepared. So that's also pretty cool. Then there's another second pathway. It's the jasmonic acid pathway, and this one is more specific to herbivores. So insects, could also be bigger, animals. They trigger usually the jasmonic acid pathway. A typical example is actually the tobacco plant. So if there's, a broader mechanical damage of the leaf, that of course also sends off this signal pathway, okay, I'm being chewed on, I'm being eaten by something, I have to do something about it, right? So that the plant then also has, certain layers of responses that it does. It first of all produces also some compounds that make it maybe more indigestible or make it even poisonous. The tobacco plant increases its nicotine production, and the nicotine actually kills off the caterpillars, the tobacco caterpillars that are eating the plant. Then also interesting, it can also be methylated. So that's why maybe also this jasmonic acid has become one of the main plant hormones for the plant to be used as a signal molecule, because this can also then transport to or be transported to other areas of the plant very quickly. It can happen within seconds because the plant is chewed on, the jasmonic acid is being increased directly, and then the methylated jasmonic acid is transported through the air to other parts of the plant or to neighboring plants. And then you have this crosstalk between plants, neighboring plants, and also microbes, because microbes can induce an immune response as well. And that's what we call the induced systemic response, so ISR. And that's actually what we want. This is a sort of preventative concept, what we use for plant protection, because we use beneficial microbes. They induce a certain response by the plant that makes it more prepared for consecutive attacks by other pathogens. That means the microbes, they don't do anything harmful to the plant. They just send some kinds of signals that make the plant more resilient against other pathogens. And that's one of the main concepts we actually use if we want to use microbes for plant protection. So I want to come to now one of the main strains that my company identified many years ago. And it's our prime plant protection agent. The strain is called FZB42. It's the FZB abbreviation goes back to the name of the research center. It's called Forschungszentrum Biotechnologie in German. So it means Research Center for Biotechnology. And it's a model organism by now because it was one of the first Bacillus bacteria that was found to be a plant growth promoting bacteria. And that already happened in the 80s. So my research center goes back to the 1950s, and they were looking in different things in biotechnology. I'll talk more about that in the second part of the talk. And they had a collaboration with the Humboldt University in the 80s, and that's where they were investigating a lot in Bacillus bacteria. So what you see here, this is an electron electron picture of the bacteria. I'll just skip that word. You see the bacteria here growing alongside the roots, and they form a biofilm, which is another layer of protection, especially against nematodes. We've seen videos of how nematodes are trying to attack roots, and they can't get through this biofilm. They just can't get through it, and they give up. Some of them actually even die because this bacteria also produces metabolites that are against, or that actually kill some nematode species. So here we used a mutant of FZB42 that included a GFP protein. Maybe you've heard of this before. This is used a lot in science to visualize where certain compounds or microorganisms, or it could be also just another protein, where it is in tissue. So you see that the roots here, so this large thing is the roots. This is a root here. And they visualized, okay, where is FZB42 in the plant? So they marked it with this GFP protein. And then you can see that it's alongside the root, but very highly concentrated at the root hairs, right? And we know that the root hairs is where we have lots of exudation and also where the plants are taking up the nutrients. So that's also very interesting. And here I just wanted to sort of summarize, the main modes of action of FZB42. Again, on the right side, you see this biofilm. You can see it. I hope you can see it on the picture a bit. It's sort of like a slight shimmer on the roots. So this is not microscopic. This is just a normal picture, but you can see it with your naked eyes. So in general, the strain can mobilize different kinds of nutrients. It produces phytohormones directly, so they influence obviously the behavior of the plant. It actually promotes the root growth. They also release precursors of other phytohormones. Then the biofilm, I already told you, protects the roots. And it can also produce specific compounds that work against fungal pathogens, but also other bacteria that could be harmful, or also nematodes. I'll go into which kind of metabolites these could be in a bit, but I just wanted to show you this diagram that just shows that the bacteria can work in a multitude of ways in helping the plants grow. And it's not only mobilizing nutrients, but it's also protecting it against pathogens. And I think you can see that here. So here I wanted to show you just what type of metabolites there are and which ones are effective against what kind of pathogen. So you have a lot of metabolites that work against fungal pathogens. So these are like bacillomycin D, fungicin, and surfactin. And you have a lot of metabolites that work against other bacteria. So you could call these antibiotics. And there are also, here's a picture showing that the bacteria, so the bacteria is here in the middle, and there's different pests on the outside. That's how you do usually like plate tests and see how does the bacteria actually work against a certain pathogen. You could also do compatibility testing like this if you want to mix different bacteria with each other, or maybe mix a bacteria with a beneficial fungi like a trichoderma. That's what we do. We do like agar plates, grow the bacteria on it, and then also combine it then with other products to see if it's compatible. And if you mix it with pathogens, hopefully you want to see something like this, where the bacteria actually fights against the pathogen and creates these zones of inhibition. So here you see it again as well, that there's a clear zone between the bacillus here and the Fusarium oxysporum. So the Fusarium is not able to breach the area where the bacillus is, and there's a clear zone between these organisms. And I added one more metabolite here. This is the certain metabolite, because I mentioned to you that the FZB42 is also effective against some nematodes, and this is the metabolite we know of that is effective and can kill nematodes. It's not every nematode species, because there's loads of different nematodes and they're quite different, but we know that it works a lot against many important plant parasitic nematodes. And this product, we mainly market it as a fungicide, but in Brazil, for example, it's registered as a nematicide as well. Yeah, I want to now come to fermentation and just give you, let's say, the general concept of fermentation. I think I don't have too much time left, or maybe I'll breeze through this. I've got until 12, right? So I just wanted to show you how fermentation works in general, and that it really depends on what kind of microbe you have, and every microbe, depending on where it's coming from, has different favorable conditions. One of the most interesting things is that we think that life on Earth is actually originated from very simple organisms like this, and they probably originated in the deep sea, because you have these geothermal vents where only some very simple organisms can survive, and from that it started this chain of events, and you have the evolution of higher organisms from there. The thing is, you can see some microorganisms can survive in very dire conditions. For us, dire. I mean, maybe for Winnipeg, not so much. But in some parts, this is very cold. I mean, in the deep sea, it's also very cold down there, and you have, methane, and in these deep sea vents, it can also be very hot, right? You've got this hot steam coming out from these smokers. They're also called black smokers, where you actually are on the other side of the spectrum, and you have like boiling water coming out at these really deep depths. And of course, because there's this huge diversity of microbes, they also have different favorable conditions in terms of pH. Right, that's what I just wanted to show you, because in the end, that's really about like the art of fermentation, getting it right to know what does this microbe need in order to cultivate it. And I wanted to just show you, or maybe go a bit over time, to show you what is actually fermentation and where is it used. You probably all know about beer, you know, and maybe cheese. But did you, for example, know that soy sauce is also made from 2 steps of fermentation? Or, I mean, there's just a list of things here, which things are made from fermentation. This is just focusing on food. You probably, I hope you can read everything, but I think you've probably heard of these things by now. I've also, like, since I'm working at the company, I've now tried natto myself. Has anyone heard about natto? Yeah, some here, very good. Maybe you've been in Japan, so it's a soybean that is fermented by a bacillus species, and I thought that was super interesting because I work at a company now that is a specialist for bacillus. So I was wondering, how come I never heard about it? And it has a very distinctive taste. It's quite strong, and you would be surprised that it's actually a soybean, and it suddenly tastes like something completely else, different. So very strong, very umami flavor. And another thing where microbes are involved in is, of course, also biofuels, things like that. People don't always think about. Lots of the industrial processes are actually involved with, or microbes are involved in these industrial processes. Production of antibiotics as well. I'll just quickly show you this as well. This is a general, flow of what fermentation is. When you grow a microbe, you have a log phase first, where maybe spores are germinating, and you start to introduce, the favorable conditions for the certain microorganisms, give it the right food. Maybe the microbe, we work a lot with aerobic microbes, right? So we need to introduce, high oxygen levels in this fermenter. So then the growth of the microbe goes up, and at some point the maximum is reached if you do it right. And then you have what's so-called the stationary phase. And depending on what kind of microbe you have, you want to maybe, for example, in our case, it's spore forming. So we actually introduce a step where we heat up the microbes to unfavorable conditions so that they create spores. Because spores are the form which we want to use as a product. They have a great shelf life, and they will only germinate when the conditions are favorable again. I think I should probably leave it at that because I'm already 3 minutes over time. Should I? Yeah? All right, then I don't want to be between you and lunch. You'll be angry at me. Yeah, I hope you enjoyed it. I wanted to show you a bit about what does it mean with concentrations, but maybe I can bring that up in the second part of my talk. I would say thank you for now, and have a great lunch.